So now we want to talk about the question of why do we have all of these introns? I mean, it's really quite amazing that we have so much apparent junk DNA that is copied into RNA and then immediately thrown away. What good are these introns? One thing that introns do for us is to allow eukaryotic cells to evolve new genes readily. And they can evolve new genes by exon duplication. So let's take, for example, the fibronectin protein. You can see here that the fibronectin protein is made up of many repeats of different domains. These domains are also found in other proteins. So for example, the yellow domain is found in cell surface receptors and other extracellular matrix proteins. The blue domain is found in blood coagulation proteins, and the red domain is also found in TPA, or tissue plasminogen activator. So the way the fibronectin was created was to take all these different domains and hook them up and bring them in from other places. And the way that that can be done is that you see, if you look now at this diagram of the fibronectin gene, you can see that each of these domains consists of either one or two exons. So the advantage of having introns in this case is that by non-homologous recombination, big chunks of the genome can be taken from one place to another, and they don't necessarily have to be hooked up perfectly because the splicing machinery hooks them up. Whereas in bacteria, where there are no introns, if there is a non-homologous recombination event to make a new protein, then it has to be perfectly in frame, and it's much harder to do because there's not the genetic space in which to do the recombination. So that is one real advantage of having introns. Now, the second major advantage we will discuss by looking at the gene number versus complexity problem. So let's consider how many genes different kinds of organisms have. So here, for example, is E. coli, a bacterium, and S. cerevisiae, budding yeast, or the yeast that is used to make bread or brew beer. E. coli is a bacterium in your gut. Now, we've already shown that bacteria are much simpler cells than eukaryotes, so you might expect that E. coli not to have as many genes as S. cerevisiae, and that's true. So E. coli has about 3,200 genes, and S. cerevisiae has about 6,000 genes. And by genes here, I'm talking about protein coding genes. Now, if we go up the evolutionary ladder, let's consider two other model organisms, C. elegans, or the roundworm, and Drosophila melanogaster, the fruit fly. Now, on the surface, it might seem that Drosophila is the more complicated organism, so it would have more genes than the roundworm, but in fact, it's the opposite way. So C. elegans has about 19,000 genes, and Drosophila melanogaster only has about 13,000 genes. So now you have to ask yourself, how much more complicated am I than a fruit fly or a roundworm? So how many genes do we expect humans to have? Or humans, mice, and even a mustard plant, so something that might be a little more complicated than a roundworm. And so if a roundworm has 19,000 genes, before the human genome was sequenced, people were thinking that humans would have around 100,000 genes. The big surprise has come that, in fact, all of these organisms have about the same number of genes, and they're all around 25,000. So it's really not a very big number. And just by this count, we're no more complicated than twice a fruit fly or about 1.3 times a roundworm. That's sort of troubling. So how can we get away with that? Well, the way we get away with this apparent lack of complexity is alternative splicing. So eukaryotes, in addition to having split genes, they defy the central dogma of biology, the original central dogma of biology. And that is that we may have one gene, but one gene can make many, many proteins. And so, for example, here is one gene where if all of the exons are spliced in, then it makes one protein. But if the red exon is left out, it makes a different protein. And then here, the blue exon, exon 4, is left out, and that makes yet a third protein. So on this particular gene, there are 3 proteins coming from one gene. And we now know from recent deep sequencing efforts that about 95% of all human genes exhibit alternative splicing. So that means that our proteome, the number of proteins that we have, is much, much larger than our genome.